Bismillahirrahmanirrahim and salam sejahtera. So today for Biology Form 4 KSSM Chapter 6 Cell Division Part 1. Before we go more further, please click like button and subscribe. Cell division. So cell always grow, divide and die. The cell die must be replaced. Cell in the body produce new cell through the cell division process. Cell division involves two stages. First is karyokinesis that involves the division of the nucleus. Second one is a cytokinesis involves division of the cytoplasm. So our cell will every day produce a new cell, maybe to replace the dead cell or to recover injury. So this process we call as a cell division. So in our body, organism cell, we have two types of cell. First is a somatic cell and the other one is gamete cell. So we look more detail what is the difference between these two. Somatic cell. Somatic cell is a body cell apart from the gamete. Beside gamete, all our cell in our body is a somatic cell. Somatic cells are produced through the mitosis process. It contains a diploid number of chromosomes. Take note the word diploid. D means double. Diploid number of chromosomes. It is each cell contains two sets of chromosomes, or we can write it as 2N. 2 means diploid. In human, somatic cell is 2N equal to 46 number of chromosomes. So human somatic cell is diploid, which is human have 46 number of chromosomes. And we go to the gamete. Gamete are productive cell. Example of gamete in human is sperm and ovum. Gametes are produced through the meiosis process. So this meiosis is the function of meiosis process is to produce gamete. It contains a haploid number. Look at the word haploid, half plot. That means half number of chromosome. It is each cell contains one set of chromosome. In human gamete, sperm or ovum is consists of 23 number of chromosome. So it's half from the somatic cell. Somatic cell have 46, but in gamete, sperm and ovum, they have 23 number of chromosomes. So we come more detail what is haploid and diploid. In diploid cell, one set of chromosome originate from the male parent, or we can say it as a paternal chromosome. Other set is from the female parent, which is known as a maternal chromosome. So both maternal and paternal chromosome have the same structure characteristic. So look at the three chromosomes without pair. So they are having its own characteristic. So this pair of chromosomes are called homologous chromosome. So homo meaning same. Okay, homologous chromosome meaning same structural characteristic. Homologous chromosome. So chromatic is chromosome. They are look like a long thread. Okay, now we are refer to the diagram. Haploid, half. Each copy of a copy of each chromosome. So we have 3 chromosomes without pair. So this one is haploid N. So the other one is diploid 2N. 2 copies of each chromosome. So one set is one set. We have a red color and blue color. So that one is 2 copies. So now we have 3 pairs of homologous chromosome. meaning one set of paternal from the paternal chromosome, the other one set from the maternal chromosome. Meaning, in our body, 50% chromosome from our father, which is paternal chromosome, and 50% chromosome from our mother, which is maternal chromosome. So cell cycle. So we going more detail about the mitosis. Cell cycle refer to the sequence of events that involve DNA multiplication and cell division to produce two daughter cells. The cell cycle consists of interphase and metaphase. So generally we can say that cell cycles only two phases, which is interphase and metaphase. But inside the interphase is made up of G1, S and G2. And it is a longest phase in the cell cycle. So in the interphase consists of G1, S and G2. Okay, so we look at the more detail about this interphase. Okay, so first of all, look at the diagram. Okay, cell cycle. So refer to the brown color of arrow. So we have two phases of the cell cycle. We have M phase, and the other one arrow is the interphase. But this interphase we have 1 to 3 stages, which is G1, S, and G2. So now we're going to learn more detail about G1 phase. So this when G1 phase start when the cell already finished the M phase, which is mitosis and cytokinesis. Meaning the cell before entering the G phase, they are already done the cell division. So in this phase, cell growing. So you know that when a cell undergoes cell division, it becomes smaller. So in G1 phase, the cell growing. So mitochondrion and endoplasmic reticulum are produced in this stage. Why? To produce mitochondrion to produce energy, endoplasmic reticulum will produce protein. Okay, in this phase. So protein also synthesized during this time. The nucleus look bigger, and the chromosome is in the form of chromatid. After they are finished in the G phase, the cell will enter to another interphase, is S phase. In S phase, we remember synthesis. S meaning synthesis. So DNA synthesis occur in this phase. The DNA in the nucleus is replicate. Okay, each chromosome multiply into two identical chromosomes known as a chromatid. So chromosome, we have the two identical chromosomes that we call as a sister chromatid. Both sister chromatid contain the same copy of DNA molecule. So they are joined at centromer. So what is centromer? Okay, afterward we will learn about the centromer. After the cell undergoes already finished the S phase, they goes to the G phase. So G phase, what happened in this G2 phase? So the cell will continue growing after the DNA develop, right? It's continue growing because it's completed the genome or genetic material, and active metabolical and cell gather energy. So this one is very active to gather energy to ensure they can enter the M phase. Okay, so we go to the N phase. Main phase made up of the mitosis and cytokinesis. Okay, mitosis is involving prophase, metaphase, anaphase, and telophase. So there is the cell cycle, which is consists of M phase and the interphase. Okay, mitosis. Mitosis is defined as the division of two nucleus of parent cell into two nuclei. Each nucleus contains the same number of chromosomes and genetically and genetic content with the nucleus of parent cell. So meaning they are produced the same identical daughter cell. Mitosis involves prophase, metaphase, anaphase, and telophase. So we can acronym it as a PROMAT. Okay. From the diagram, you can see this is the photo of the tip of shoot of a leaf. Okay. You can see all the four phases in this cell. You can see anaphase, telophase, prophase, and metaphase. So now we're going to learn more detail about the each stages in mitosis. So remember the word PMAT. So the first one is prophase. Pro mean awal or earlier. Okay. So here, what happened to the cell? Chromatic start to shorten and thicken. So because it's become shortened and thickened, so we can see it through the light microscope. The chromosome made up of two identical traits called sister chromatic. So it's developed into two. So you can see the shape like a butterfly. So both sister chromatic are joined at the centromer. Okay, now we learn about the centromer. So look at the diagram, the red color chromosome. So the center thing, the center of the chromosome we call has a centromere. So it's come from word center. Okay, centromere. So this chromatic attached at the centromere, and also centromere provide place for the spindle fiber to attach to it. The nucleus membrane disintegrate, the nucleus disappear. So this nucleus disappear to allow the movement of the chromosome to all the part of the cell. The centriole move to the opposite pole and the spindle fiber start to form. So you can see the centrioles start to move to each pole. Okay. Then go to the PMAT M. M metaphase, or remember the word M is middle. So what actually happened is in this phase, centrioles are at the pole of the cell. The spindle fiber maintain the chromatic at the equatorial plate. So meaning all the chromosome will be aligned themselves at the middle of the cell plate. The chromosome become aligned in a single row. Take the word here, single row on the equator plate. Metaphase end when the centromer begin to divide. So the centromer, you remember the sister chromatic attached at the centromer, meaning the centromer able to split into two. So third phase is PAMAT, anaphase. Remember the word A, away. So in this stage, the centromer divide into two and the sister chromatic is separated. Okay. Spindle fiber shorten, contract, and the sister chromatic are attached to the opposite pole. So this you can see the sister chromatic move toward the opposite pole. Anaphase end when the chromatic arrives at the pole of the cell. Okay, you take note. Spindle fiber always attached at the centromer of the chromosome. So the word away here show the sister chromatic will move, split away, goes to the opposite pole. Okay, so we go the next last page of the mitosis is telophase. PAMAT. T, the word two. Remember word T? Two. When the chromatic are at the opposite pole, they are now called the daughter chromosome. So chromatic arrive at the opposite pole, we call it as a daughter chromosome. So each pole contains one set of complete and identical chromosome. So there we have a two set of chromosome, right? One of each pole. Chromosome are shaped again and fine chromatic thread, and nuclei are formed again, and spindle fiber disappears. So now you can see under microscope the nucleus. A new nucleus membrane is formed, and telophase stage is followed by cytokinesis. So now what is the difference between mitosis and cytokinesis in the animal and plant cell? After mitosis, we have next stage is the cytokinesis. So first of all, we look at more detail about the animal cell. Cytokinesis actually different between animal cell and plant cell. So in animal cell, cytokinesis is divided of the cytoplasm that happen immediately after the nucleus is formed. So after telophase, the new cytoplasm, the cytokinesis will occur. The plasma membrane constructs in the middle of the cell between the two nuclei. Microfilament, you can see in the diagram, right? The microfilament at the point of construction will contract, so it will cause the split. The cell membrane will be split to form two daughter cells. Then you got two identical daughter cells. So now we come more detail how this cytokinesis in the plant cell. In the plant cell is formed after these two nuclei, two nucleus develop, two nucleus membrane develop. So it will form vesicle combine to form cell plate at the center of the cell to separate between these two nucleus. The cell plate are surrounded by a new plasma membrane. A new cell wall substance is formed among the space of the cell plate. So there is the space between the cell plate. So it will form a new cell wall. The cell plate expand outward until they are combined with the cytoplasm. Cellulose fiber are produced by the cell to strengthen the new cell wall. So you can see now two diploid daughter cells are formed which are identical. So what is the necessity of the mitosis? Okay. First, for embryo development and organism growth, mitosis ensure the rapid cell growth occur. So girl, if you still remember when you are newborn baby, your weight must around 2 or to 4 kilogram. But now you are already 50, 60 kilogram, right? So there is the increasing in number of cells. So this one, because of your cell undergoes mitosis. So this is the first necessity of mitosis for the organism to grow. Second one, for the regeneration. So if you experience to catch a lizard, right? So when you hold the tail of lizard, it will break. So don't worry because it able to regenerate a new tail. So this one also will undergoes mitosis. Third one, when the body injured, so I'm sure everybody experienced the injury, right? So after a few days, your worm cured. Okay, actually to cure the injury, your cell have to undergo mitosis to replace cells that are died or damaged. Okay, another necessity of cell is for the asexual reproductive. Normally this one in the unicellular organism such as bacteria, okay, fungi. So mitosis at organisms such as hydra, in this diagram, hydra, to produce new individual through the formation of new bud. So to form a new bud, this part of organism will be undergoes mitosis to form a new organism asexually. So another necessity of mitosis, you look at the first diagram, the culturing technique using stem cell from animal which are then cultured in the laboratory to produce meat. So this is one example of the cell division. So we take the cell, and the cell will be multiplied, mitosis, to form a new meat, to produce meat. Okay. Second diagram, stem cell therapy use stem cell from bone marrow to treat the damaged cartilage. So this one also the necessity of mitosis. Last one is the tissue culture. We want to get a quality plant in the large amount, okay, in the short period. So we are using a technique of culturing plant tissue is used to produce young plant through the culturing of parent cell without going through the fertilization process. This one also involves mitosis. Okay, that's all for today. Thank you for watching, and please don't forget to subscribe. I'm Cikgu Ros from SMK Convent Ipoh. Bye.